This is the Chapter 5 lecture video where we will talk about the 4 organic molecules: carbohydrates, lipids, proteins, and nucleic acids. Macromolecules are polymers that are built from monomers. A polymer is simply a long molecule consisting of many similar or identical building blocks that are linked by covalent bonds. Each of the small repeating units are called monomers. However, as we will see, some monomers are able to function on their own without being bonded to anything else. Before we talk about the 4 macromolecules, we want to talk a little bit about how they are made and broken down. Both of these processes are facilitated by enzymes that help speed up the chemical reactions. When monomers are connected, it is called a dehydration reaction. 2 molecules will be covalently bonded to each other with the loss of a water molecule. Each monomer will contribute part of the water molecule, so one will contribute the hydroxyl group and the other will contribute a hydrogen. And that reaction will be repeated over and over until the polymer is complete. The breakdown of polymers is called hydrolysis, which is essentially the reverse of dehydration synthesis. Here, the bond between monomers is broken by the addition of a water molecule. So we think about digestion. Hydrolysis is used to break food down, and dehydration synthesis is used to build the molecules that our bodies need. Here you can see a picture example of both the dehydration reaction and hydrolysis. Each cell has thousands of different macromolecules, which will vary between cell types and individuals. However, they are all constructed from 40 to 50 common monomers. The first macromolecule we will discuss is carbohydrates. Carbohydrates are sugars and polymers of sugars. A monosaccharide is the simplest carbohydrate, which can also be called simple sugars, and is the monomer for building complex carbohydrates. A disaccharide is 2 monosaccharides put together, and polysaccharides are lots of monosaccharides put together. All carbohydrates are composed of carbon, hydrogen, and oxygen in a 1:2:1 ratio. That means that they will have the same number of carbon and oxygens and double that number of hydrogens. Monosaccharides are joined by glycosidic linkages, which is a type of dehydration reaction. And structurally, they can either be linear or branched, depending on the type of sugar it is. Glucose is the most common monosaccharide and is one that can function on its own without being connected to anything else. In water, it forms a ring structure, so that's how we find it in living things. They have a carbonyl group and multiple hydroxyl group. You will see that most sugars end in the 3 letters OSE, but that is not always the case. Cells are able to extract energy from glucose during cell respiration by breaking it down and then eventually being able to form ATP. The simple sugar molecules are a major fuel for cellular work, and their carbon skeletons are raw materials for synthesis of other small organic molecules like amino acids and fatty acids. The sugar molecules that are not immediately used are incorporated as monomers into disaccharides or polysaccharides. For example, plants transport carbohydrates from the leaves to the roots in the form of sucrose, which is a glucose molecule linked to a fructose molecule, and later will be broken down and used individually. So here you can see 2 examples of disaccharide synthesis via dehydration reaction. Polysaccharides are polymers that can contain a few hundred to a few thousand monosaccharides. They serve as storage material and are hydrolyzed as needed. So that's like one function of polysaccharides. Plants store starch and animals store glycogen. Both of those are a polymer of glucose, but their glycosidic linkages are in different places. Polysaccharides can also serve as building materials for structures. For example, cellulose is part of the plant cell wall, which is yet another polymer of glucose, and chitin is found in arthropod exoskeletons and fungi cell walls. A third function of polysaccharides is determined by the sugar monomers and the position of the glycosidic linkages, which you can see here. So those are three different polymers of glucose: starch, glycogen, and cellulose. So even though it's all made up of glucose, those glycosidic linkages are in slightly different places. And here is another comparison of those, looking at starch and cellulose. Next, we will move on to discussing lipids. Remember that hydrophilic means attracts water and hydrophobic means repels water. This class of macromolecules does not include true polymers like what we see with the other three, but they are all grouped together because they all mix poorly with water. They consist mostly of hydrocarbon regions, where we, just have carbon, hydrogen, and lots of both. They vary in form and function and include waxes, some pigments, fats, phospholipids, and steroids. Some other examples of lipids you might be familiar with are triglycerides and cholesterol. Fats are assembled from smaller molecules by dehydration reactions, and they are constructed from two kinds of smaller molecules, glycerol and fatty acids, which you can see in the example to the right. Glycerol is an alcohol, and a fatty acid has a long carbon chain. And all of the nonpolar carbon-hydrogen bonds in the hydrocarbon chain are what make the fat hydrophobic. Fats can be saturated or unsaturated. A saturated fatty acid is saturated with hydrogen, which means there are no double bonds between carbons. Fats made from saturated fatty acids are called saturated fats, and we're describing here mostly animal fats that will be solid at room temperature. Diets rich in saturated fats, however, can contribute to cardiovascular disease. Unsaturated fatty acids have one or more double bonds between carbons, which creates a kink in the carbon chain. This group is referred to as oils, so think about plants and fish, and they are liquid at room temperature. One of the major functions of fat is energy storage. They can store twice as much energy as polysaccharides. Here are examples of saturated and unsaturated fats, and you can see on the unsaturated fat where the double bonded carbons are, it creates that bend or kink in the hydrocarbon chain. Phospholipids are one type of lipid that we will spend some time talking about relatively soon. They are a major component of the cell membrane. They're similar to a fat molecule, but it only has two fatty acids attached to glycerol. The hydrocarbon tails are hydrophobic, but the heads are hydrophilic. And because of that is why they arrange in a bilayer. So we have the hydrophilic region on the outside and inside of the cell, and the hydrophobic region trapped in between that. When added to water, phospholipids do self-assemble into a bilayer simply because of that hydrophobic and hydrophilic property. Steroids are another large group of lipids, and they are characterized by a carbon skeleton consisting of 4 rings. And they are distinguished by the chemical group that is attached to the ring. So the example to the right is cholesterol, which is crucial for animals. It is a component of the cell membrane and is a precursor for other steroids, meaning that it can easily be modified to be turned into something else. One of the interesting things about cholesterol is that it's made in the liver and obtained by your diet, but your body makes enough cholesterol for you. So anything that you're consuming outside of that is simply extra that you have. Next, we will move on to proteins, which are the most abundant and diverse group of macromolecules. Nearly every single function of a living thing depends on proteins, and it accounts for more than 50% of the dry mass of most cells. Some functions of proteins include that they speed up chemical reactions. So, there we're talking about enzymes. They're used for defense, storage, transport, cell communication, movement, and structural support. Here are some picture examples of those functions. Most of these we will talk about at one point or another in this class. Cell communication is one of my favorites because I think it's super cool. All proteins are constructed from 20 different amino acids that are connected by a peptide bond. So amino acids are attached, we have a peptide bond. Therefore, a polymer of amino acids is called a polypeptide. And then a protein is the functional molecule made up of one or more polypeptides that's going to be folded and coiled into a specific 3D structure. The monomer of proteins is called an amino acid, and they all share a common structure. They have an amino group, which is that part containing nitrogen, a carboxyl group, which is the COOH, and an R group that are all around essential carbon. The R group or side chain is different for each amino acid, and whatever the R group is determines that specific amino acid's properties. So whether it's polar, nonpolar, has a charge, is hydrophilic or hydrophobic. Here are the 20 amino acids with their R groups. So what's the same for all of them is in purple, and then the different colors tell you the property of the amino acid. I do not expect you to know all of these, but I do think it's super cool. So let's talk a little more about polypeptides and polypeptide chains. Amino acids are joined by a dehydration reaction, and the resulting covalent bond is, as I said, called a peptide bond. And this will happen over and over again until the polypeptide is complete. So this could be a few amino acids to 1,000 or more. Each polypeptide has a unique linear sequence that comes from whatever the DNA molecule says. One end of the chain has a free amino group, which is called the N terminus, and the other has a free carboxyl group, which is the C terminus. Next, we're going to talk about protein structure and function because they are super picky. So the specific function of proteins is a result of the 3D folding of its polypeptides. Protein specific structure determines how it works. Because proteins almost always, their function depends on its ability to recognize and bind to some other molecule. So the example here is an antibody protein that needs to be able to bind to a protein from the flu virus to help us with for our immune system to do its job. There are 4 levels of protein structure. Primary is simply the sequence of amino acids. Secondary are the regions stabilized by hydrogen bonds between atoms of the polypeptide backbone. Tertiary, 3D shape stabilized by interactions between the side chains. And quaternary is only when the protein consists of 2 or more polypeptides. For more details on this, you should refer to figure 5.18 in your book. So what determines protein structure? Its chain and 3D shape. Some folding will occur as the protein is being made, but sometimes, particularly the big ones, will be sent off to other parts of the cell for modification. And it also depends on the physical and chemical conditions surrounding the protein. So changes in pH, salinity, or temperature can weaken the bonds and interactions between the protein, which leads to an unraveling and losing its shape. And as we know, when a protein loses its shape, it cannot do its job. And that process is called denaturation, which is it's biologically inactive. Sometimes they can renature, but not always, which you can see in the figure to the right. Last, we will talk about nucleic acids, which are polymers of nucleotides. There are two types of nucleic acids: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. They are both important for gene expression. So DNA provides directions for its own replication to make sure that DNA is in every cell, and directs RNA synthesis. And then RNA controls protein synthesis. But we will talk about that much, much later. There are three components of a nucleotide. They all contain a 5-carbon sugar, a nitrogen base, and a phosphate group. A nucleoside refers to the nucleotide without the phosphate group. Each nitrogen base has one or two rings that include nitrogen atoms. There are two families of that, pyrimidine and purine. A pyrimidine is one 6-membered ring, so we're talking about cytosine, thymine, and uracil. And purines are a 6-membered ring fused to a 5-membered ring. Those are adenine and guanine. DNA contains the nitrogen bases A, G, T, and C, and RNA contains A, G, U, and C. DNA contains the sugar deoxyribose, which is the same as ribose, but it is missing an oxygen atom on the second carbon, and RNA contains the sugar ribose. Adjacent nucleotides are joined by a phosphodiester linkage, which is once again a type of dehydration synthesis, and the phosphate group that links the sugars between the two nucleotides leads to the sugar phosphate backbone that we see with DNA, not with RNA because it only is one single-stranded. The ends of the polymer are different. DNA does have a direction. The 5' end is what is attached to the phosphate group, and the 3' end has a hydroxyl group. During DNA and RNA synthesis, nucleotides are only added to the 3' end, which does lead to some problems, but we will talk more about those when we get to DNA replication later in the year. Here you can see the structure of DNA and RNA molecules. DNA is a double helix, meaning it has two strands. One runs 5' to 3', and the other runs 3' to 5'. That's why they're anti-parallel. These strands are complementary, not identical. The strands are held together by 2 or 3 hydrogen bonds, depending on whether it is a CG or an AT. Remember that A pairs with T, and those are held together by 2 hydrogen bonds, and C and G are held together by 3. RNA, on the other hand, is single-stranded, but complementary base pairing does occur sometimes with tRNA. It'll loop back on itself. And that is all for chapter 5.